Welcome everyone to today's HPL seminar. Today we'll be hearing from Stephanie Ross. Stephanie is joining Walter Herzog's group as a postdoc later this year in September. Stephanie earned her bachelor's and PhD in biomedical physiology and kinesiology from Simon Fraser University. Her PhD was advised by James Wakeling, and her PhD project title was The Effects of Muscle Tissue Inertia on Contractile Performance. For a little less than a year, she has been a postdoctoral fellow at the University of British Columbia in the Rehabilitation Research Program. There, her research has focused on the impact of stroke on skeletal muscle behavior. Dr. Ross's research interests include using experimental measures of muscle across scales and muscle models to improve our understanding of muscle mechanics in health and disease. In her free time, Stephanie enjoys downhill skiing, distance running, and exploring the outdoors with her dog, Briar. I'll now turn it over to Stephanie to start sharing her screen for her presentation, and then we'll have a question and answer period to follow.
Awesome. Thanks so much, Emily. I'll just share my screen here. Then can you see my slides or the notes?
Perfect, with no notes.
Okay, great. All right. Hello, everyone. Thanks, Emily, and for the introduction, and then thanks everyone for having me here to do this talk. So today I'm going to be talking about my research that I've done that uses a combination of experimental measures and then also model simulations to understand muscle mechanics in both healthy muscle and then also muscle that's been affected by different diseases and conditions. Okay, so in terms of the overview, I'm going to cover some work that I did in my PhD in James Wakeling's lab at Simon Fraser University, where I examined the effects of inertia due to muscle mass on contractile performance. And then I'll talk a bit about a study where we used one of the models that I developed in my PhD to understand the impact of altered tissue properties that we see in cerebral palsy on overall muscle function. And then I'll talk a bit about the work that I'm currently doing as a postdoc at UBC in the Rehabilitation Research Program based at GF Strong Rehab Center, where I'm looking at the effects of different tissue property changes on muscle function following stroke. And then throughout the talk, I'll kind of cover a bit about what my vision is for the future of these different lines of inquiry. And then at the end, I'll briefly touch on some of the work that I might be doing when I start as a postdoc in Walter Herzog's group in the fall. Okay, so I probably don't have to sell the importance of understanding muscle to this audience, but in case some of you are still not sold, I'll briefly cover kind of why we should care about muscle and how muscle works. So to start, muscle is incredibly important, and it's what drives the motion of our bodies and allows us to interact with our world around us and the environment. So everything from getting out of bed in the morning to an animal escaping prey, skeletal muscles are kind of what are behind that movement. And then in terms of specific applications of knowing more about how muscles work, greater understanding could lead to better models to predict body movement. And so we can do things like predict the outcome of surgical interventions and things like that. Then it could also improve our ability to mimic the action of muscles in things like active prosthetics and exoskeletons. And then it's also important for identifying targets for interventions to treat different diseases or conditions that affect muscle function. Okay, so one of the gaps in our understanding of how muscles work that I explored during my PhD research was the effects of muscle inertia, or the fact that muscle tissue has mass, and that mass accelerates when muscle contracts. So most of what we know about the function of muscle comes from studies on single fibers or small muscles from animals like mice and rats. And then we just scale up that understanding and assume that muscles in larger animals like humans and elephants behave the same without considering the effects of that additional tissue mass in larger muscles. So this assumption that greater mass doesn't really matter is reflected in how we model the behavior of whole muscle across animal sizes. So you can see on the image on the right is a typical Hilltype muscle model. And in those models, we assume that whole muscles can be represented by the properties of just a massless single fiber. So these models typically don't account for any effects of tissue mass. So the question is that a reasonable thing to assume? So are the effects of muscle mass in larger animals like humans and elephants really negligible, like we assume they are? Even though those muscles are so many times larger than the fibers or the small muscles from animals like mice or rats. So to explain a bit about how greater mass in larger muscle would create, would have an effect on muscle. So if you scale up the size of a parallel fiber muscle, like on the left, by a factor of 2, like we have here. So this would result in muscle lengths, velocities, and accelerations increasing by a factor of 2. And then because a muscle's force scales with the cross-sectional area, which would increase by a factor of 2 squared in this situation, the forces will increase by a factor of 4. Then the muscle mass is proportional to the muscle's volume, and the volume will increase by a factor of 2 cubed. So that will result in an increase in the volume by 8 times. So for a larger muscle, this one where we have the scale of 2, but even if we isometrically scale up the size of a muscle, the mass of the muscle and the volume is relatively larger than the muscle force that's available to accelerate it. So just to reiterate, the inertial effects of muscle mass are typically never accounted for in muscle models of any size. But this kind of logic here is why we would expect that the effects would be even greater for human muscles and muscles in larger animals. Okay, so to explore the effects of muscle mass on contractile behavior, during my PhD, I used a Hilltype muscle model that I developed during my undergrad honors project in James Wakely's lab as well. And then this model accounted for the dynamics of tissue mass. So it's actually the one that's on the bottom of these two. And so the tissue mass is accounted for in a series of distributed point masses along the length of the model. And then the one on the top is kind of a typical Hilltype muscle model, like it's a simple representation of muscle that doesn't account for any effects of muscle mass. And I use that model kind of as a comparison to see what the effects of mass were relative to that typical massless Hilltype model. So I wanted to develop a system that would allow me to examine what the effects of muscle mass are on muscle behavior without having to constrain the kinematics or dynamics of the muscle. So I developed this modeling framework that was composed of a damped harmonic oscillator in series with the Hilltype muscle model. So this setup allowed me to cyclically contract the muscle while providing only an activation trace. So like the one you see in the top in green. And that activation trace just drove the dynamics of the muscle, created muscle forces, which moved the harmonic oscillator, and that created cyclic patterns of contraction. So then I optimized the oscillator properties so that the muscle would contract and change length, because if you just randomly select properties, in a lot of cases, the muscle just doesn't really move or isn't able to contract. And then I also determined the scaling factors that would ensure dynamic similarity of the harmonic oscillator muscle system to keep the relative effect of the oscillator constant across muscle sizes as I scaled up the size of the mass enhanced muscle model. So then I ran simulations of both of these models, the mass enhanced Hilltype model that's on the bottom, and then the typical massless model within the oscillator system. And then I also ran some additional simulations with sinusoidal length changes. So I prescribed the position of the end of the muscle to follow a sinusoidal trajectory. And that kind of mimicked the experimental setup that you would see kind of in situ experiments when you have a muscle motor that's driving the length changes. And then comparing the simulations of the mass enhanced model to the massless kind of gave an idea of how the effects of mass would impact muscle behavior during these types of cyclic contractions. Okay, so just to kind of explain how I got calculation. So I looked at the effects of work per cycle or mass specific work per cycle. And so for those of you who are kind of aren't familiar with what a work loop is, so that's just when we have these cyclic patterns of contractions. And then we plot the muscle force over the muscle strain, and we get one of these loops that we have here that we refer to as a work loop. And then the area inside of the loop represents the work per cycle. So for a larger muscle, so when I scale up the size of any muscle model whether it has mass or not, or if you have a larger real muscle compared to a smaller one, you would expect that the work would be larger because the cross-sectional area and therefore the forces and also the lengths are larger. So in order to separate out the size effect, like just the fact that we increase the size and the forces and the lengths are larger from the effects of the inertial effects, the effects of the tissue mass, I normalize the work by the kind of theoretical mass, which is a bit confusing, which is the volume times the density to determine the relative effect of inertia due to the greater mass. So we get this mass specific work per cycle. And in the massless model where there is no inertial effects, we see and would expect to see that the mass specific work per cycle is constant across sizes. So here are some of the results from the simulations that I ran with the two different models, and then also from scaling the mass enhanced model up in size. So going from top to bottom, we have the mass specific work, and then we have the mass specific metabolic cost. And then on the bottom is the efficiency. And these are all expressed as a percentage of the massless Hilltype muscle model. And then on the left within each panel is the maximum excitation for a given simulation. So it's the maximum was 20% on the left or 0.2, and then 100% or maximal on the right. And then in the different colors, so there's the mass enhanced, in the dark blue, there's the mass enhanced model that accounts for the inertial effects. And it's kind of the smaller size model. And then the lighter blue is that same model scaled up in size by a factor of 10. And then the horizontal red line is the massless model. So we can see here that the scaling up the mass enhanced model decreased the mass specific work per cycle. And that effect was greater when the model was larger, as you can see by the blue being lower, the light blue being lower than the dark blue. And then it had very little effect on the metabolic costs that I predicted from using different metabolic cost models that have been reported in literature. And then in terms of the efficiency, it primarily just followed the trends with the work per cycle. So overall, so increasing the mass of the muscle resulted in lower mass specific work, approximately the same metabolic cost per cycle, and then a decrease in efficiency. So those results were cool to see, but the muscle models that we used, as with any mathematical model, is just a representation of the actual system. So I thought it was important to be able to confirm that these effects, these inertial effects, were actually occurring in real muscle. So during my PhD, I visited Andy Winner's lab at Concord Field Station at Harvard to do a study on rat muscle in situ, where we added different magnitudes of mass into the muscle. So this is kind of the setup for doing that and achieving that. So I made this system where a rod rotated about a pivot. And then on one end of the rod, there was a pin that I inserted into the middle of the muscle. And then on the other end of the rod, I attached weights of different sizes to add effective mass to the muscle acting through the pin. And so that pin was inserted into the rat muscle. So here it's experiments rather than models. And so then I cyclically activated the muscle by sending signals through the nerve that innervated the muscle. And then I also constrained the muscle model to follow a sinusoidal trajectory using a servo motor. And that was to create similar contraction cycles to the ones that I previously simulated in the 1D model. Okay, so here are some of the results for the work per cycle. So again, the plot on the left shows the results from the experiments. And you can see that the greater added mass resulted in lower work relative to the unloaded condition where we didn't add mass to the acting through the pin. And then when we simulated those same contraction cycles on the right with the 1D mass enhanced Hilltype model, we saw similar reductions in work per cycle. So while these in situ, these in situ kind of results kind of give us a bit more confidence that those 1D models were behaving kind of as expected. So while that was nice to kind of see, we still didn't really have a full picture on how muscle mass alters the behavior of real 3D whole muscle, because we only added mass to a single location in the in situ muscle. And then in the model, it was just one dimensional. So we still didn't know how the 3D structure and having distributed mass in all dimensions would affect muscle performance. So that brings us to the next study. So for this study, I examined the effects of tissue mass on muscle performance in 3D. So to do that, we developed a fully dynamic 3D continuum model, or a visco-hyperelastic model, which accounted for the effects of distributed tissue mass. And then this model is made up of 1D fibers, which you can see as the red arrows. And then those fibers have the same intrinsic properties as the 1D Hilltype model, but they're embedded within an isotropic base material or background material that represents the connective tissue, blood, and other fluids kind of surrounding fibers in real muscle. So then we ran some simulations, I ran some simulations with the muscle geometry of about the approximate size of a human calf or plantar flexor muscle. And with the same work loop contraction cycles as the previous studies. And then I scaled the model up in size by a factor of 2.5 and also 3.5 to see what the effects of muscle mass were across a range of muscle sizes. So in the figure on the bottom right, we have the results for the mass specific work per cycle. So on the vertical axis is the work as a percentage of the scale 1 geometry, so the kind of base geometry. And then on the horizontal axis is the scale. So the different lines are different pronation angles, but regardless of the pronation angle of the model, all these simulations showed a reduction in the mass specific work per cycle when muscle size was increased. So then to make sure that manipulating the muscle mass was resulting in similar changes in tissue behavior for the simulated 3D muscle as real muscle, I compared the tissue accelerations in the X direction, or the along the muscle length direction, across the muscle model during simulations, the 3D simulations, and I compared that to the same kind of parameter output as the in situ rat plantaris muscle from the previous study. So for the in situ muscle on the left, I looked at the acceleration at the point of the pin insertion, where I added the effective mass. I used a LED photodiode pair to measure kind of track that position over time. And then for the graphs, the difference in the max acceleration is expressed as a percentage of the unloaded condition with no added mass for the experiments. And then the scale 1 geometry for the simulations. And for the 3D simulations, I use this point just kind of in the center of the model to track that over time. So for both the in situ muscle and the 3D model, we found that there was a larger difference in acceleration at the different locations with greater mass. So the accelerations have units of 1/2 which controls for the fact that the point at the end is at a longer length than the point in the middle. So if you have a muscle where the mass is negligibly small, then you would expect that there would be little or no difference in the relative accelerations at different locations. So you'd expect just kind of a flat line if there was no kind of effect of inertial effects acting to change the accelerations in the center of the tissue. So here we found that the accelerations are different and they change with mass in a way that's similar to for both the in situ muscle and also the model simulations. Okay, so overall, in summary, muscle mass has important consequences for muscle function based on the results of my PhD thesis. And accounting for inertial effects and increasing muscle size results in lower mass specific work per cycle, Approximately the same metabolic cost per cycle for the contractions that we looked at, and also lower efficiency per cycle. And then this has, as I mentioned kind of earlier, this has implications for how we potentially model muscle and represent them in whole body musculoskeletal models, and also how we mimic the behavior of muscle in prosthetics and exoskeletons and things like that. Okay. So in terms of a few ideas that I've had kind of to move forward with this work from my PhD thesis, one thing that could be done is to examine the effects of muscle mass on contractile performance during activities of daily living. So while I examined the effects of, I examined mass during cyclic contractions and time varying activation, I always prescribe the activation with a repeating square wave that I ran through an excitation activation transfer function. So it was pretty idealized. And then in terms of the muscle lengths, I used either the harmonic oscillator or prescribed sinusoidal length changes to the end of the muscle, both of which probably don't reflect the full range of, well, we pretty much know they don't reflect the full range of contractions that occur in vivo in real muscle. So to do this study, I would first collect motion capture, ground reaction forces, and EMG from people doing a range of different activities. So you can see the kind of setup that I did for a previous study with cycling on the left, but it would be kind of similar setup. And so they would do activities like walking, running, jumping, sit to stand, kind of a full range of different types of contractions. And then I would use that data to run it through simulations of a full body open sim model to get the predicted lengths and excitations, and then use those real predicted lengths and excitations from the open sim model to see what the effects of muscle mass are, and to drive those previous 1D models to get an idea of how muscle mass affects contractile performance during a range of more realistic kind of contractions. And then another thing that could be done, which is kind of not all that exciting, but is I think a good next step and kind of necessary, would be to validate the forces of the 3D dynamic model against in vivo forces from something like a rat muscle during gait in vivo. So to do that, I would probably, I would like to conduct surgery to insert radio opaque beads into the muscle. And then that measures with kind of CT scan arms to be able to measure the deformations of the tissue during in vivo contraction. And then connect a strain gauge, or what we call a tendon buckle, to the tendon to estimate the forces of the muscle. And then when the animals recover from surgery, you can get them to run at different speeds on the treadmill and at different inclines to get a range of muscle contractions, and then see how the muscle is behaving as the animal is moving. And then I would run comparable simulations of the 3D model to those in vivo contractions, and then compare the forces of the model to the in vivo forces from the real rat muscle. Okay, so in addition to improving our understanding of the mechanisms that underlie muscle function, one of the benefits of the work in my PhD thesis was that I ended up developing kind of a whole suite of different muscle models that can be used to probe other questions. So in this study that was led by a master's student, Ryan Kano, we used the quasi-static muscle model to examine the relative effect of some of the mechanisms that are known to vary with CP muscle compared to that of muscle from typically developed kids. Okay, so when we're looking at the properties of typically developing muscle versus muscle that's been affected by CP, there have been experimental studies on kind of tissue biopsies that have been collected from kids during surgery that have found that sarcomere lengths are longer in CP muscle, as well as there's also more extracellular matrix. So the ECM fraction or volume fraction is larger. And there's also more stiffness of that tissue. So that surrounding tissue, tissue surrounding the fibers that are in pink. And then there are other factors that are kind of experimentally known to be different between CP muscle and typically developed muscle, like, for example, titin content. But for this study, we just focused on these different factors here. So then Ryan ran some simulations of the model and varied these different tissue properties to see their relative effect on the passive force of the block of muscle. And so, yeah, so he varied, we used a quasi-static model and then used blocks of muscle to represent the tissue biopsies. He varied the sarcomere length, ECM volume fraction and stiffness, and then also measured kind of the passive force length properties. Okay, so now some of the results that are published in this paper here for the effects of ECM volume fraction and sarcomere length. So the plot on the left shows the results from when the simulated muscle had a volume fraction of 5%, which is about the approximate range for a typically developed muscle. And then on the right are the results from when the model had a volume fraction of 40%, which is kind of more representative of muscle affected by CP. And then within each plot, the different colors show different sarcomere lengths. And the black one, where there's arrow pointing to it, that represents sarcomere properties of typically developed muscle. And then in the pink, or kind of coral color, it shows simulations with the longest sarcomere lengths, where the sarcomeres have higher passive force when the muscle or the block of the muscle is at resting length. So as you can see from changing the sarcomere length, so as you can see from these plots, changing the sarcomere length had a greater effect on passive stiffness when the ECM volume fraction was low. Which you can see by there's kind of greater spread between the different lines on the left compared to on the right. And then the relative effect of, and then relative to increasing the sarcomere length, the increase in ECM volume fraction or the kind of total ECM content appeared to have a greater effect on the overall passive muscle stiffness in comparison to changing the sarcomere lengths, which you can see kind of in the slope of the left plot versus the right versus the spread on each one. Okay, so then from there, I kind of had the idea to use the same model, that same quasi-static 3D model, to examine the effects of different tissue properties and muscle properties in a muscle that's been affected by stroke. So that's kind of what I've been doing in my postdoc at UBC since I started last September. So there are actually a number of similarities between muscle that's been affected by CP and muscle that's been affected by stroke. So in paretic muscle or muscle affected by stroke, we see decreased cross-sectional area and volume, increased tissue stiffness, increased fat accumulation, and also altered patterns of regional activation. So the plot that's on the bottom left is from a study that they looked at the shear wave velocity of the passive paretic, non-paretic, and then control muscle, kind of as a proxy for measuring muscle stiffness. And they found that the paretic muscle had higher shear wave velocity relative to non-paretic and control muscle, indicating that the tissue was likely stiffer when the muscle was paretic. And then these colored maps on the right show regional activation from high density EMG measured from the bicep muscle in people who've had a stroke. And as you can see from comparing the paretic plots on the top to the non-paretic on the right, that the different sides in a given person, this is from one representative participant, the different sides had different regional patterns of activation as measured by the high density EMG. So for my current postdoc work, I'm collecting both ultrasound and high density EMG from people who've had a stroke on both their paretic and non-paretic sides. And then in terms of combining the model and experiments, I'm using the same quasi-static model, but with geometries that are taken from MRI scans of people with stroke, so their paretic and non-paretic sides. And then I'm varying the different tissue properties like the fat, the tissue stiffness or the ECM stiffness, and then also the regional activation using the high density EMG that I measured in the model. And then once the simulations are finally done, I'll compare the relative effect of these different properties on the muscle force. And then to validate the model, I'll also compare the results to the measured ultrasound measures and then kind of place the experimental results within the full parameter space. Okay, so I don't really have any results from the simulations yet, but I do have some kind of interesting ultrasound videos to give you an idea of how paretic muscle can behave relative to both non-paretic and control muscle. So on the left is my experimental setup. So I mounted a portable force plate on the wall, and then I had participants, or I'm currently testing, I had participants sit in a chair with their foot kind of on the plate, and then I get them to contract their calf muscles to reach a force target that I specified based on what their maximum voluntary force is. So I do maximal and then also submaximal, and then I do force matching as well as effort matching on the paretic versus non-paretic sides. And so in this picture on the left, this person just has the high density EMG on measuring their medial gastroc during the contractions. Okay, so on the right is some ultrasound images. So just to kind of orient you to what's going on here. So on the top of each image this is the surface of the skin, and then this would be their superficial aponeurosis, if you can see my cursor. Let's see. And so the top is the superficial aponeurosis, and then this is the muscle in contraction. So this is a maximal voluntary contraction, and you can see for this control participant, there's it's fairly easy for the fascicles, or those kind of white lines, which are bundles of fibers, to rotate as the muscle contracts. And then, so this is then these two videos on the bottom are for the same individual that has hemiparetic, so affecting one side primarily compared to the other, chronic stroke. So this is the non-paretic side. So it's behaving kind of similarly to the control participant, but we do see a bit of like hyperechoic. So it's whiter and it shows up a lot brighter than the control participant, indicating that there's probably a bit more fat and a bit more connective tissue in that muscle. But then when we look at the paretic side, we see some pretty noticeable differences. So you can see it's very difficult. The fascicles barely kind of rotate. And you can just see based on the way the muscle is kind of shaking a bit that it's quite difficult to perform those contractions. And in most of the individuals I've tested, there's quite a difference in strength on their paretic versus non-paretic side. So you can really see kind of striking differences between those two sides. Okay. Okay, so one of the potential future directions for this work is I'd like to do kind of a longitudinal study to examine the time course of muscle changes post-stroke. So currently in my study, I'm looking at people who are subacute, so they've recently had a stroke and are currently inpatients at GF Strong Rehabilitation Center. And then I'm also looking at and testing people who are in the chronic stage. So it's greater than 1 year, but the average of my participants so far is about 7 years post-stroke. So these images are just ones that I've collected actually from different individuals. And you can see that there's kind of a degradation across the images to the point where on this one here, you can't even really differentiate what a fascicle is from surrounding tissue. And it's the muscle is quite atrophied. It's very hyperechoic. It shows up almost white, indicating that there's a lot of fat and connective tissue. But it would be really interesting to be able to see how within one individual, or a group of individuals, but having themselves as controls, is to track that progression of muscle changes over time. And then it would also be great to be able to collect MRI with diffusion tensor imaging to be able to quantify 3D changes in muscle structure over time with the progression of the muscle changes. And then another kind of future direction that I would like to pursue with this work is to kind of look at different studies, or to look at studies examining muscle changes in a rat model of stroke. So rat models of stroke are used far more in kind of the field of motor control to understand how stroke is affecting kind of movement patterns and things like that. But what I would like to do is kind of do studies that you wouldn't be able to do in human participants because of potentially ethical issues and also difficulty in controlling different lifestyle factors and things like that. So one thing I would like to do is to be able to look at the effect of stroke on muscle tissue properties and protein content and things like that. So here we have some images, example images of different stains to look at the fat content and also ECM content. So it'd be interesting to see in when a stroke is induced in a rat on their paretic versus non-paretic side, how do those tissue properties change? And because this would be difficult to do in humans who've actually had a stroke. And then Botox is a common intervention to treat spasticity post-stroke. So kind of inappropriate resting activation of the muscle that causes a lot of discomfort and challenges with mobility for people. So I would like to be able to see what the impact of Botox is on muscle that has been affected by stroke and how that varies over time, kind of in the rat model. And then also something that would be interesting is to look at the impact of stroke on in vivo muscle behavior during gait. So similar to the previous study validating 3D dynamic model, to be able to use the radio-opaque beads and CT scans in real time to be able to measure how the muscle is behaving on the paretic versus non-paretic side and how that differs from rats that have had induced strokes versus those that haven't. Okay, and then in terms of some of the work that I could potentially be doing in Calgary in Walter Herzog's group when I start in September. So early in this talk, I kind of talked about the simulations that Ryan did with the 3D muscle model, examining the effects of sarcomere length and ECM volume fraction and stiffness. And so this is kind of carries on with some of that work in that area of research. So it would be kind of working with some of the people who in Walter's group who are already working on these things, so kind of to test the effects of reduced tighten in CP muscle on active force. So confirm the reduction in tighten content in muscle that's been affected by CP. So these are from tissue biopsies that have been taken from kids that have had different types of surgeries, so like tendon elongation surgery. And so confirm the reduction in the tighten in the CP affected muscle in comparison to control muscle. And then also test the active myofibril force. And then also test the effects of collagenase on the tissue behavior. As I mentioned, Botox is something that's commonly used to treat increased stiffness of the muscle. But there have been some studies where they've shown that Botox can actually degrade the tissue and increase the fat accumulation and the passive stiffness of the tissue over time. And then there's also some studies that show that not all people who have what is deemed clinically as spasticity actually have inappropriate resting activation. So it might be that they wouldn't even benefit from Botox injections. So one of the things that could instead be used is to use collagenase to break down the collagen or the ECM matrix. And so potential things would be applying controlled doses of collagenase and testing passive vascular force and the protein content. And then also using that previous 3D muscle model to run complementary simulations and vary the different tissue components to kind of expand on the previous findings from the quasi-static model. Okay, so that's all I have. Thank you so much for your attention. And thank you to all the contributors of this work and all the people who have helped me along the way, and also the funding sources. Thanks.
Thank you, Steph. At this time, I'd kindly ask the audience members that if you're comfortable to please turn on your Zoom camera to allow for a better discussion. To ask your question, you can use the raise hand function under reactions on Zoom, or you can type your question in the chat box and I will read it out loud. Art, please go ahead.
I think this is getting at something that you're already intending to do, but the question is about the effect of mass which I certainly agree that there should be some sort of a cost for moving the mass of muscle. But do you have any idea or expectation of what you're going to find for sort of whole body movements? How much of the effect there would be, and maybe to go along with that, in which task do you expect the effect of moving the mass of the muscle to be the greatest?
So I think in terms of, so if I were to compare the mass specific work per cycle again, I would expect that, given that for the, say the sinusoidal contractions that I did, the strains were intended to kind of mimic what we would see in vivo contraction cycles. So they were kind of in a realistic range. So just based on driving those model simulations with those in vivo contractions, say during walking or running, I would expect kind of a similar range. And then in terms of what would be the greatest effect, I think so the ones that we're considering doing, so we have, I mentioned sit to stand, running, walking, and then jumping. So I would expect kind of jumping to be the highest because the driving forces or the accelerations of the tissue are greatest. And then potentially running would be next, walking. And then for close to isometric contractions, I would expect the effective mass to be the lowest. And then I didn't really kind of go into it in this talk, but one of the kind of long-term things that would be cool to do is to be able to integrate it actually in the effects of mass actually into like a whole body model. And there I would expect that that the predicted excitations would potentially be lower. So rather than, because in the simulations that I ran, the activations and the length changes are always controlled and are always prescribed. And so in like a simulation, a whole body simulation, that wouldn't be the case. So if you're predicting the activation, so I would expect that it would require higher activations for a larger muscle to get the same work per cycle or the same forces, just to overcome the effects of that mass, essentially.
I see. Well, maybe it's a question of just what you divide by in order to get an indicator of the cost. Where I think here by mass specific, you're considering how much of the cost of shortening is due to moving the mass for that muscle. But my question about everyday movements or just about any kind of movement is that I'm not sure how much of the cost. So let's say sit to stand. Certainly when I sit to stand, I use my glutes and the glute muscle moves and the mass moves. But I feel like my sense is that the big mass that I'm moving is the body, and I'm lifting the glutes, but that's different from the question of how much energy is spent on basically, you could say, accelerating the mass of the glute along their line of action. So I'm just wondering if you have any idea of how much of the total energetic cost of, say, a sit to stand would be attributable to the particular mass effect that you're talking about.
Yeah, that's a good question. I don't really have a good sense of what it would be for the total relative to other factors. Yeah, I don't have a really great sense, but that would be something to for sure pursue.
Yeah, it would be really interesting.
Yeah.
Yeah, thanks.
Thanks.
Walter, go ahead.
Yeah, thanks a lot, Steph. I have had a couple of really fairly simple questions, I guess. And the first one is, I just wanted to go back a little bit to, the very beginning when you say, force increases by cross-sectional area to the second power, and volume and mass increases to the third power, the scaling that you had there. And it did seem to me, if I understood it correctly, that you used like a cylinder type of thing to represent the muscle. Is that correct?
So like a Hilltype muscle model technically doesn't have shape or volume. It's but like to be able to get the maximum isometric force that we scale by the cross-sectional area, and then also the length. But so for the visualization, I use a cylinder, but it could be represented by anything with the same cross-sectional area and length.
Yeah, I was just wondering, because it seems to me that probably that scaling how mass and force scales, I would assume, will probably depend on really the structure of the muscle. Because if I imagine that I have a cylinder like you had, and now just I make the cylinder twice as long, then I have twice the mass, but the cross-sectional area would be the same. So the force would not have increased at all. There would be no change in cross-sectional area. And then the other scenario would be where I have a pinnate muscle, and let's assume the pinnate muscle is so long that if I cut all the fibers perpendicularly, I get exactly the length of the muscle. So then if I then add that same length to the muscle without changing the thickness, then I would also have twice the volume, but I also would have twice the physiological cross-sectional area. So in that sense, the physiological cross-sectional area, depending on the structure, would change very differently, and the force would change very differently depending on what structural arrangement you assume the muscle has. And I was wondering if you considered that, for example, when you did work on the plantaris of the rat, which I assume is a unipinnate muscle. Was that ever a consideration, or was that just kind of a not looked at?
So I guess in terms of kind of your first point about like if you add the same block later to make it longer. So based on the scaling of, say, the 1D or the 3D model, or probably also real muscle, if you increase the size or like scale it in the way that we did, it ends up being like a length dependent effect, like you say. So it ends up being the inertial effects are dependent on the length scale with the length squared, because the increase in the cross-sectional area is also represented in the increase in the volume. And so if I double the length of the model, it doesn't, or it has an effect, whereas if I double the width, it doesn't have any effect, and the work per cycle is the same. So that's a totally fair point. And then in terms of the pennation angle, so for the 3D model, I did look at geometries with, I think it was 4 different initial pennation angles. And so I found when I ran the same simulations, having greater pennation angle, like being a more pennate muscle, resulted in smaller mass effects, essentially. So as I scaled it up in size, there was a smaller reduction in the mass specific work per cycle when the muscle was more pennate. And so, which is kind of interesting because there is some scaling, like experimental studies that have shown that when they measure muscle architecture across animals of different sizes, or like lizards of different sizes, that there is like a positive scaling relationship with where pennation angle increases with muscle size. So it potentially, I mean, it's kind of unclear at this point, but it potentially could be something that's a mechanism to mitigate the effects of the greater mass, like in a larger muscle.
Yeah, no, that's good. I think that would be an interesting thing to potentially look at a bit more carefully. The other thing that I was wondering, when you showed the results with your work loops, and then you added mass to the muscle, different masses, and then your energy inside your work loop, your work performed, decreased, and I assume that was what you plotted there was the force measured at the tendon, kind of integrated over the displacement, over the shortening and the elongation. Is that correct?
Yeah.
Yeah. So if you now added to that the work performed because you added the additional mass, would you have then gotten the same amount of work all the time? So, or in other words, did you get the total work always about the same? And then if you replace the mass effect, then you got the decrease in the tendon force that you measured?
I'm not sure. I think I would have to think about that one.
Okay. Yeah. Yeah, but yeah, okay, good. And yeah, I think I let other people talk. I might have another couple of questions later on, but let's give other people a chance.
I'll jump in with a question, Steph. I just have a methodological question. How do you track fascicle dynamics in some of those ultrasound images that you said have a lot of fat, and it's like really hard to discern individual fascicles?
So that is a challenge. So the one image that I showed, which was the very atrophied one, that was kind of when I showed the time course, and it was at the one at the end. Like that one, I wouldn't be able to track the fascicle behavior. I would expect that even if I could, it wouldn't, the muscle really didn't even move when the person was doing a contraction. And so for that data, that would probably be excluded from my data set in terms of validating the muscle model to it. But I do think it's kind of has been interesting to see the different kind of presentations on the ultrasound of the muscle, and kind of to recognize the limitation of using the ultrasound, because we can't really measure things in individuals with quite a lot of atrophy. And then also in other conditions besides stroke, so things like spinal cord injury. I know there's people, I have colleagues who have tried to do ultrasound in people who have had complete spinal cord injuries, and the muscle is so atrophied that they can't really, it just really doesn't show up as much.
Mm-hmm.
So, yeah.
Are there similar challenges with estimating the architecture from DTI images of those atrophied muscles too? I'm not as familiar with that.
I'm not sure about that. I haven't done DTI, use that measure myself, but I think I would guess that it would. I think someone might be more of an expert on this than me, but from what I understand, DTI, diffusion tensor imaging relies on the fact that there's the diffusion gradients are different across the fiber or fascicle versus along. And so I would expect that if those fascicles or fibers are really atrophied to the point where they've been replaced by a lot of fat or connective tissue that doesn't have that anisotropic diffusion, then it would be kind of the same issue. Yeah.
I'll maybe jump in with one more question too. Sure. Is there any scenarios when the inertia of the muscle mass could actually be beneficial in terms of energy expenditure, be helping the individual during a certain locomotion task?
Yeah, that's something that is interesting, and I feel like I've had. That was something I think that came up a while ago. That's ringing a bell. I think we had talked about it potentially being something as a power amplifier during, say, jumping and things like that. But we don't really have. I just did the cyclic contraction, so for the contractions that I did, it didn't have any helping effect, but potentially in other conditions, since we didn't measure a full range of contractions that are possible.
Walter, go ahead.
The other couple of questions that I had was, when in your model, when you have fat infiltration or ECM or fibrosis, yep. And let's say for simplicity, you have 95% contractile material. Now, half of that contractile material is replaced either by ECM or by fat. Does that mean that your force goes down by 50% as well? Or is there a certain interaction effect there that since you change the structure of the muscle, that might actually help preserve some of the force or reduce it even more than you would just expect?
Yeah, so I'm trying to think back. So there was one of James's previous PhD students did look at the effect of intramuscular fat on the muscle. And he looked at different tissue for kind of regionalization of that fat. And so when it was distributed, it had a different effect than when there was a lump in the center of the model or whatever. because it affected how the fibers could kind of track around it and things like that. But in terms of like the way that the volume fraction of the ECM and also like a volume fraction of fat is represented, it does scale down like the fiber forces then.
So it kind of goes, you kind of linearly down.
Yeah.
Yeah.
But to what oh, sorry, go ahead.
No, go ahead.
But yeah, I was going to say, I don't like for the contractions that we did, we only did, like Ryan only did simulations of passive contractions or passive length changes, I guess. And so it's not clear kind of what interactions effects there would be with the active force, active fiber force with the changing the volume fraction. Yeah. But I would expect that it would be purely like based on that ratio because of how like complicated and three-dimensional the model is.
And so in your model right now, if I understood that correctly, then if let's say 10% of a contractile material gets replaced by ECM or by fat, in terms of the force output, that would be identical, that would be the same.
Not necessarily.
Okay.
So if it were, say, like a 1D model, then I think that would be the case.
Yeah.
But because of the three-dimensional and like nonlinear behavior, and it's prescribed at each point, like there's thousands of points within the total model, that I don't think that it would be identical. It would scale that way, scale linear.
The other thing, go ahead. Yeah.
I was just gonna say I haven't done those simulations to kind of confirm that, so kind of guess.
And the other thing that I thought was really nice was, when you showed, I think it was ultrasound images and how they were distinctly different, in your, I think it was a stroke model, where you assumed that there might be more fat there. I was just wondering, because a lot of people these days seem to be interested in the quality of muscle, in obesity, in chronic diseases, cancer and other things. And I was wondering, have you ever tried to somehow quantify from your image density of the ultrasound and then actually check what the fat infiltration was and what kind of a relationship there might be between what you see on the image and the actual fat content or the actual fibrosis ECM content?
Yeah, I haven't from my images, but I think that is something that people have done where they quantify like the amount of kind of white spaces in the muscle. Yeah, I think it might be difficult with my images, or I guess I would be concerned that there would be other factors that are affecting the amount of echo intensity, other than just the fat content and ECM. So things like I have, I use a balloon filled with water that's in between the ultrasound image and the surface of the skin to increase both the kind of resolution of the images, but also to increase the distance, just like a camera and ultrasound image has like a distance to focus. And so that kind of increases the distance. So I'm able to focus on the medial gastroc, which is really superficial in comparison to like something way deeper. So I don't know to what extent that is also impacting that intensity and if that would vary with different tissue qualities and things like that. So, but it is interesting to be able to,
So, and then the last question I had there, the longitudinal data that you showed us, I think it was from a stroke patient, you said, is that correct?
So that would be kind of theoretically what I would maybe expect from if I'm
that was not real data.
No, so those are different from different people that I measured.
Okay, yeah. So you have not done like a longitudinal thing, yeah. So that deterioration seemed to be quite big, in terms of the different intensities that you measure there. How long do you think that might take if you followed a stroke patient that had
That's a good question, and it's would make.
Are we talking weeks or months or years or?
So I definitely noticed a difference. So in people who had a stroke, say a year to 2 years ago, I still see definitely a difference between their paretic and non-paretic sides in terms of like the intensity, the echo intensity, like the whiteness, and also even just like the thickness of the muscle and how it behaves. But so that person where it was very atrophied, I think had a stroke 18 years ago or something like that. But that individual was also kind of a specific case because she was only ambulatory in her small apartment and primarily used an electric wheelchair. So like that kind of thing would also contribute. But I think that's kind of very extreme, and you would still see changes like in a couple of years, I would expect. And then probably I would expect a lot of people don't even get to that point if they're still like mobile, and still a lot of the people that are doing different types of activities and exercise programs and stuff. So yeah.
Okay, great, thanks.
Thanks.
Any other questions?
You have a very small and shy group today, huh?
Final questions for Steph? Okay, great. Please join me in thanking Stephanie Ross for her presentation today, and thank you to the audience members for the questions. I hope to see everyone again next week for the HPL seminar, where we will be hearing from Fabian Hoytz, and his presentation title is Layer-Wise Relevance Propagation for Running Biomechanics.